नंदिनी आपने ज्वाइन किया। नंदिनी आप सुन पा रहे हैं मुझे?
हाँ जी सर, हाँ जी। हाँ जी।
ठीक है, ऑडियो वीडियो दोनों ठीक है आपका? ऑडियो दिख रहा है, वीडियो हमें
सर
दिखाई दे
एक्चुअली
रही
मेरे पास कुछ लोग टेक्स्ट कर रहे हैं, उनके पास लिंक नहीं पहुंचा है। तो एक
है।
बार
अच्छा
मैं
हाँ
लिंक
ठीक है,
शेयर कर
भेज।
दूं?
ठीक है, भेज भेज दीजिए, भेज दीजिए।
सर 35 पे शुरू करें ताकि और
ठीक है,
ज्वाइन
ठीक
कर
है,
ले।
ठीक है, ठीक है, ठीक है।
ओके सर, ओके।
नंदिनी अब स्टार्ट करिए, आप पार्टिसिपेंट्स ज्वाइन करते रहेंगे, वो तो 6:40 7 बजे तक ज्वाइन करते रहते हैं।
ओके सर,
ठीक है,
ओके।
हाँ अब आप स्टार्ट करिए।
Good evening, everyone. This side Nandini Sharma from Agni Faundation, Bharat. and I welcome you all to the Day 6 of Faculty Development Training, Annadata 2025. It is my great privilege to welcome our eminent speaker for today's session, Dr. Sunita Chaudhary, scientist at ICRISAT. Dr. Chaudhary is widely recognized for her work in crop physiology, plant breeding, and climate smart agriculture, where she has been consistently contributing towards building a sustainable future for farming. With over 8 years of rich experience in both public and private research organizations, she has played a key role in bridging scientific innovation with practical solutions for farmers and researchers alike. She has authored more than 22 research papers and 3 book chapters, and her impactful work continues to guide and inspire scholars across the agricultural community. Beyond her publications, what truly sets her apart is her commitment to mentoring students and young researchers. Having successfully guided many students, she has shown us that true leadership lies not only in advancing knowledge but also in empowering the next generation to dream bigger. Today, she is here with us to share her insights on AI technologies for accelerated crop improvement and climate resilience. This is a subject of immense significance as agriculture adapts to the challenge of climate change and the need for technological advancement. We are eager to learn from her vast expertise, her vision, and her experience. Dr. Sunita Chaudhary, ma'am, aura reflects humility, wisdom, and inspiration. Her presence today reminds us that learning has no limits and innovation in agriculture knows no boundaries. On behalf of everyone present here, I extend a heartfelt welcome to you, ma'am. We are honored and grateful for your time and guidance, and we look forward to a truly enriching session. Thank you so much. Over to you. The session is yours.
Thank you, Nandini, for beautiful introduction. It's very motivating. Good evening to everyone. My name is Sunita Chaudhary. By training, I'm crop physiologist. Over time, we also venture into advanced technology, which can be used for physiological way of phenotyping. So the way physiologists study about abiotic stresses, it doesn't serve the purpose to breeder. Breeding has a volume, so they have 1,000 plots to evaluate. And with climate change, this all traits like drought adaptation, heat adaptation, nutrition challenges they have to address. So in this context, we started venturing into the trait we study. What are the proxies? What are the sensor or advanced technology which we can use, which can help breeder to rapidly assess their line, generate data over that, phenotype them, and make a best selection. So today my presentation is more concentrated over that what are the advanced technology available, and if we can avail them or if we can develop in-house facilities for that. So I will not go more on physiology science, but the technology which is available and how AI is helping agriculture. So some of the key technology which we work at ICRISAT, I have documented for presentation. And I'm very glad to be here. And thanks AgriMeet Foundation for the opportunity to share my knowledge with wider audience. If my slide is visible, can I start the presentation? Yeah.
Yes, please.
Yeah. Yeah. So as we can see that most of the people are from breeding background. So we all know that we make crosses and we have to advance up to 6-7 generation, and that there are a lot of numbers. And how many crosses will you make, that the volume you have. Most of the selections or data generation is very manual. So let's say a lot of technology has come, but how many technology we are using in core R&D of the agriculture? Let's say, have an example. If I have to measure plant height, what option I have? I just take a scale and go to the field or send our people to the field. There is not much technology which we are using. Thinking in this direction, we started working. What are the integration we can do with technology which can actually help breeder in R&D rather than the product development, final product. So we realized that actually the phenotype is very complex because it's a result of many things. Genetics itself doesn't express as such the phenotype. Phenotype, the outer appearance of plant or the performance of plant depends on many things. Its genetic is one thing, but the environment is another, management is other, and the interactions of everything. That's why you see the one of the India map on the other side. You see the potential. It's not like it's all about genetics of the lines you're breeding. It's also the environment. Sometimes your environment is constraining. So if your environment doesn't have potential, no matter the best material also, if you put in that environment, it's not going to give the outperform or the yield which is like equivalent to the favorable environment. So the red color represent that how much stress is there. So that kind of stress where the potential of your genetic, like the same material, if you plant in somewhere in deep soil, good water conditions, favorable environment, assure irrigated condition, and if you plant it the desert areas of Rajasthan, it's not going to give the same yield. Everybody is aware of that. So environment also has a constraint, and keeping that in mind, what traits can fit there? Like what? Let's say the example in pearl millet, if we go for assure irrigated condition, mostly monoclump, single stem is more successful. All the hybrids are based on the single stem. But if we go to A1 zone, which is the most driest zone of pearl millet, that more tillers are helping. Like tiller, all the land races are more tillering. So tillers are giving better yield than monoclump. So each trade has a trade-off based on the environment. So environment define the production also. So we cannot be only on genetics. We have to also consider the environment. So to deal with the complexity, agriculture need technology. So breeding needs support of this knowledge on environment, this knowledge on management, knowledge on genetics, everything. So we try to use this AI-based technology, which can help them to overcome some of the constraints and give them better insight. So I'm documenting today 3 technology. One is LazyScan, which is phenotyping technology, is a phenotyping platform. Second is UAV technology, is new science and how we can use for advanced application. Third is nutrition. So in nutrition, what are the sensors? What are the AI we are using? It's about that. So as you know that it's this slide would mostly are used for wider audience where people don't know about the breeding process, but I think our audience knows. So I'll skip that. So for selection, you need a lot of traits, and all the traits measurements are very manual and very time taking, labor intensive. So as the phenotyping is important tool, we have to also work on the precision of and technology supports, which can accelerate the crop improvement. And genetic gain, if you see the formula of genetic gain, it's also depend the how precise is your data. That also help in achieving the precise genetic gain. So it's not like technologies are not there. If you go to European market, there is $1 billion technology available, which is very robotic, very cobotic types, which can go to field. But are they suitable for our environment? Whether they can serve the purpose what we need it. Our land holding size research plots are very much designed in different way. And most of this technology doesn't work for us, and expenditure is also very high for this kind of technology. So um, when it comes to AI and technology, market is full of products. While choosing, my recommendation is like you look for fit for the purpose. Do not fit your requirement to that product, but see what is you need and whether that product has the capability to serve your purpose. Rather, sometimes fancy equipments are installed, but they are nominal and they are like for showcase purpose. The actual breeding programs, it's not serving. So my strong recommendation is like always need-based technology. We should look for the vendor which has capability to co-develop. Do not go for precise like very finished product where you don't have a scope to customize it. It should be sustainable, precise, and cost effective. So some of the phenomics facility for climate smart crop, what we have at ICRISAT, you are seeing the aerial view of that. One is LizzieScan, which is like high throughput phenotyping platform. You can see the blue one in the middle one, which has like 5,000 microplots, which get characterized for 15 traits in just 2 hours. So it's very, very high throughput. Second is the lysometric facility, which you are seeing covered with the rainout shelter facility. So you can do on-season trials here. You can impose the stress in season, and it can protect from rain, and your stress performance still can be evaluated. A great example of that, sometime back I was working for a seed company. So whenever we put a stress trial, always rain come, and that it doesn't get evaluated. So we started taking the trial in off season, in post rainy season, but the performance of that post rainy season never matched to the actual environment. So then companies started sending their material to Chile. Chile is a country in South America, which is at the very down. And in rainy season, the chances of rain is like less than 1%. So that's the best environment. But you see how much pain it is there to send the material and cost. Companies can afford, but whether our R&D, Indian R&D can afford, no. So the best solution is this kind of rainout shelters and with lysimeter system, which can give you very classical and precise results for drought screening. Third one is the field you are seeing. So it is laser leveled, precisely boundary and GCP equipped field, which we are using for UAVs, drone based phenotyping for field conditions. So a little bit brief about LizzieScan. So LizzieScan is an automated 24/7 monitoring system where we have the sensors. So sensors are RGB, NIRS, and also the LiDAR. So it scan the plants and it make a virtual image. And then the AI algorithms are set to extract the trait. It is so fast that per second, per microplot, it obtained 40 to 60 images to form the virtual image, which we call 3D point cloud. It moves over 4,800 microplots and generate data very fast. It's less than 2 hours. So let's say 1 hour, 120 minutes, yeah, even less than 80 minutes is for a whole platform. So it's quite fast. It's a canopy dynamics, all those transpirational dynamics. So specifically built for component traits of adaptation, which help us to study the environmental stress and what are the traits which are underplaying for those adaptation. So since you have a big high throughput phenotyping platform, it also spit a lot of data. So handling data is also important. Otherwise, the high throughput will be has a bottleneck of data analysis. So we automated that high data analysis also with IIT Bombay, and that pipeline also set. Then it became a really high throughput where you can deliver data in just few days. So just one short video I shot personally with my drone over the LizzieScan. So you can see when under Chickpea mission project, we have 5,000 germplasm lines from gene bank, and those has been screened here. So 5,000 screening in field may take 2 to 5 hectares depending on plot size. But in LizzieScan, we can do very easily this. As a very large platform, it is unique in the world. In open condition, this big automated large platforms are not available anywhere in the world. So as I explained, it has RGB, red, green, and blue, and also NIRS based. It has a dual scan, which forms very dense point cloud over the dense point cloud. When I say, it means the virtual image of the plants. So scanners are very, very powerful, very adaptive. It's you can see the 3D point cloud. How does it look like? And the sensor scan. So these are the traits which it measures every time. So every day we have these traits get measured. In fact, it runs 24/7. So every day some 10 to 12 data points we obtain over each microplot. So we are very closely every day we are monitoring the growth of the plant. which if you try to do in field, it will be very destructive. Every day you have to harvest, so that's that many plot you have to grow. But this is like very automatic. So we get biomass, we get height, we get leaf area, we get leaf angle, projected leaf area, inclinations. We also have a spectral rate because those scanners are very high throughput. Having a nurse, so this help us to study disease and all this. So this is just a glimpse of this 3D point cloud traits we are working, and point clouds are always repositories. So we are continuously developing different AI based models, CNN based model to extract more traits. Suppose I don't have right now algorithms for tiller, but I'm working on it. Maybe tomorrow I'll get it. So whatever the old experiment I have already conducted, I have a repository of that point cloud. I can still go back and extract those data. So I need not to repeat my experiments. This also has the facility of load cell, which every 15 minutes it monitor the water loss from the microplot. So that is also giving a quite number of insight how the adaptation traits are working, what is the transpiration rate, what is the transpiration efficiency of the plant. So a lot of things we are able to do with this single platform. Data analysis, as I mentioned, we have to handle those complexity. So for analysis, what pipeline we have developed, it has like outlier removal, missing data imputations, and that spat analysis, which will little bit smoothen your data, and you can see everyday basis how the growth is happening. This is again the imputations pipeline. So this IoT is helping us in pre-processing. You can see the raw data is very scattered, but then the data quality assessments we do. We also select a window where the genetic expression is high and where genotype to genotype comparisons can be facilitated, because it's capturing data every day, so you don't need to have a comparison every day. When initial days when genetic expression is less and the genotype to genotype differences are very less, we may not need to select those windows for the comparison. So we select critical window for that. We do pre-analytics and also phenotype clustering we do. So this is something very brief examples of this chickpea wild crosses. So you see the domesticated chickpea species, which is in blue, has a linear regress growth. The growth rate and the early vigor is very, very straight because it has been domesticated over 5,000 years. But if you see the wild type and semi-domesticated one, initially it grows very slow and then it pick up the biomass. But the wild type is always has a very slower growth rate. So growth rate defines most of the things. Your yield, your adaptation to different stress is very much dependent on the growth rate. So this machine is under those underlying traits, which we call component traits, is able to capture it. One of the great example of this machine where under one project, Dr. Vashni group has done 7 seasons of testing in 5 locations, and they mapped the QTL hotspot in chickpea. And when we use the same material in LizzScan, we found that in 5 weeks of assay, we mapped the same QTL hotspot. So this is the efficiency of machine, like where you can cut down a lot of time and also the resources which has gone in 7 seasons of testing in multiple locations. This can give. Always the heritability we get with LizzScan is higher than 80%, like for most of the traits, except if the traits are not those critical. Second example, where we have characterized all the mini core collection, germplasm collections of finger millet for vigor, BC NAM population, all those big population which numbers are in thousands, it get characterized in less than month or 2 month time. Like it doesn't take much time, and you see the population, each population identify as a differently. Second one example of in pearl millet, where water uptake we are monitoring. It has been done with lot of gravimetric method. So you have to weigh every 2 hours those pods, how is the water loss happening in this plants. But if you see these load cells are giving the purpose, like it automatically it is measuring. So we map the same LG to Qtel, which is mostly responsible for drought adaptation. It has been revalidated. So the efficiency of this platform is quite good. We can also plant health, we can monitor very easily because those spectral traits are there. We may not be able to see this with visible eyes because our eyes, human eyes, see in the visible range of 400 to 700. But this platform has a capacity of NIRS, which is like a higher band, 1100 to 2500. Multiple spectrals are there. So that can see the plant health much before our human eye can see. So this is also we are using for this disease and nutritional trait detection, those deficiency. This is some of the sterility mosaic disease quantification. So hue is the one of the spectral trait which we are exploiting for disease identification. So drought, this is the kind of transition we are giving with this technology. A lot of hard work for root study, for those senescence study, disease study, and yield adaptation study. This can be easily done with this kind of technology. This is only possible because this is just a machine. It formed the 3D point cloud. It also formed the images, spectral it's capture, but the underlying technology is AI, which translate your spectral information, your image information into data. So this is like the technology is always there, but until you apply the AI to translate into data what you need, then it become like advanced or it's so AI is in background of many of the technology which is helping us in. Second, like although we have beautiful phenotyping platform, but all the material um, uh, all the material can come here, but it will not give the actual environments. So we have to go to actual environment where UAVs are the best vehicles. So UAVs can take your sensors to the field or the location where you want. So this is one of the important UAV we don't see as a high throughput, but the sensors what you are carrying or the way you are using UAV, that will give you an advantage. If you want to use for spraying, it's just a mechanical use, but this technology can be used for more advanced purposes. So where you can hang the sensors and take it to the field. So can we digitalize the field phenotyping in actual location rather than bringing every material here? Or wherever this high throughput phenotyping platform is there, that has its own advantage. But when it comes to actual environment performance, we have to take our sensors to the field. So in this direction, there were a lot of legislative restrictions over UAVs, but it has been released somewhere in 2020. And 2021, we started our UAV initiative where we initiated for field phenotyping. We collaborated a lot of with IT institution. So IIT Hyderabad, IIT Bombay, IIT Indore, and IIT DM Kurnool. These ITs are like our partners, and they help us to how to drone only capture the images, but how to translate it to data. They help us in applying those AI-based algorithms. So one of the key message which I always give through our presentation, when we use UAVs plus sensors, it can be used for very mechanical advancement like spraying, surveying, monitoring. But this technology has a lot of more potential. Because of restriction, we didn't advance much. Since this technology has come, a lot of startups have come in this business, and we are progressing. But we are still not up to the level what the outside world is there, but we are progressing. My always recommendation is to look for the advanced application of UAV, which has a huge potentials, and it can bring proper like transition in agri system rather than just a mechanical advancement. So you can see you can hang the sensors whatever you want to use for field phenotyping. That also I will come to that. But for optimization, you need a very precise field experiment, at least for 2 seasons. So the algorithms can be set and it can be validated. Once your algorithm pipeline is set, then you can take your UAV with sensors to any field and you can phenotype those fields. So a few traits which we have initially optimized on two of the model crop. One is sorghum and another is groundnut. So you see that we selected intentionally the genotype which has a huge variation for plant height, for biomass, and also for nitrogen uptake. So and second level of variation we created in our data point by giving the external treatment. So the first block you are seeing is for the nitrogen, which has a proper fertilizer and proper irrigation has been given. Second block, what you are seeing is having well watered. We have given sufficient irrigation, but nitrogen has been not given. Very minimum doses of nitrogen is given just for a good establishment. And third one you are seeing where nitrogen is given, fertilizer is given, but water has been stressed out. So you see that this also creates level of variation in your pipeline. When you are setting the algorithms, you should set it or create a variation which can capture. So tomorrow, if you want to phenotype in a marginal land where the performance of plant is very, very marginal, then your algorithm should still work. Otherwise, if we only develop on good plants, probably your algorithms will not work for the stress environment. So this is one we have done. Second, we groundnut sorghum is a model crop for all long stature crops like cereals like sorghum, pearl millet, finger millet, maize. Second one is groundnut, which is representative of short stature plants like chickpea, groundnut. And here we have we cannot give any nitrogen stress because they are legumes. They fix their own nitrogen. So we have only given well water and water stress. And we have fly the drones. You have to use GCP, RTKs, all those protocol you have to follow. We develop the SOPs also. So this is the level of prediction we are able to make. So plant height, the algorithm which is developed and predicted and compared with the actual measurements, you see we have a 95% correlation, which is good enough. We don't need 100%. 95% of accuracy is like good enough. You fly one time and you have your plant height without manual investment and cost investment. Canopy cover, we have 19. Biomass, we have 92. So all the dual purpose crop development, always you have to harvest 1 by 1 meter of plot, dry it, take the biomass. So you can select for high biomass, high grained kind of lines. And one of the important achievement is like the nitrogen content of standing crop we were able to assess, and that has a correlation not as good as plant height, but it is 90%, which is I still value it because this is good enough. For groundnut, it was a bit difficult, but we used the hybrid AI model and then it worked well. So we have 83% of accuracy. We are also working with other traits. So once this pipeline is set, we are overspilling on 10 crops. Now we have 6 legumes and 4 cereals. So sorghum is there, maize is there, pearl millet is there, and finger millet is there. And same level of nitrogen I have stressed. So these are the validation and overspilling. Once the algorithm is set, your AI technology started working for model crop, you can overspill on other crops so that it can serve the wider purposes or more of the breeding programs. So basic pipeline is that we are using RGB. Most of your traits can be optimized with RGB. It's a simple camera. Analysis is also simple. But if your traits are a little bit complicated, you can use multispectral. And if it is more complicated, like early detection of water stress, nitrogen stress, hyperspectral can be used. One of the caution in this, like as you complicate your sensor or advanced sensors you are using, multi or hyper, your analysis is also going to be tenfold complex. So more advanced sensors, more complex analysis. If you are not ready for that, then our principle is first to use RGB. If it doesn't work, then we advance our analysis pipeline. We use more of AI. If that also doesn't work, then only we move to multispectral and hyperspectral. And trust me, a lot of things can be easily done with RGB. We don't need those advanced sensors until your purposes like those traits are really complex. In hyperspectral, it captures the images, the spectral information in bands. You have cubes. So for each channel, there is 100 of cubes in that. So most of your time goes in dimension reductionality, means the removing the extra information which is not required for you to analyze. So this is something, a question on use of sensors. We usually, if I have to, let's say if I have to measure plant height, I choose the time during flowering, because most of the expansion vertical growth happens till flowering. If I have to take senescence, I take near maturity. So these are the different plant growth stages where we should capture. We have developed the SOPs for that. Then we also, same time, we take the ground truth, we take the light, and then we predict. This is in discovery. So, and then we validate. If it is good enough, we validate one more season, one more field, and if everything work well, then we launch the algorithms for blind predictions. Because this capture a lot of data, you also need a cloud-based storage system where you can have the way to analyze it, and high performance computings are required. So we also collaborate with a lot of drone startups, which are every part of the world, every part of the India. Now we have more than 1 lakh startups registered in India. So wherever our multi-location trials are there, we just give them SOP and they get the images. So this is basic pipeline. We give importance to validation. Sometimes when we work with IT people or startups, they don't do validation. They are so confident on their algorithms that they think it is predicting 100% accurately, but in actual scenario, it doesn't happen. So always emphasize when you are working on UAV-based pipeline, at least first year or second year or 2 season data, you must validate with ground truth. If ground truth is not done, you are always in a risk of wrong assessments. So this is the pipeline hours where you see is mostly for IT people, where we use different methods, which is AI-based methods. This is the one which I was saying for nitrogen content of the. So you can see how the non-AI models didn't give the enough accuracy which was required, and hybrid AI models have the advantage, gave the better advantage. This kind of technology can be used for multi-location trials. India is far behind, but I'm very hopeful that in 5 to 10 years, we'll be able to solve this kind of purposes. Automations, once you develop the algorithm, it's not enough, you have to automate. So automations will give you another level of advancement where everything, you don't need an IT person to run your algorithms. It can be translated into a software where you input your images and then you have the data. So to reach that stage, it will take time, but if you see Hyphen, those are these like phenotyping industry, they have done it. Like they have a chlorophyll Pix4D, where you just input your images and it will give you data. These are some of the exploration area which has been not exploited so far through UAVs and has a potential that it can help. So as we can see, there is a lot of scope with UAVs, and maybe in future we are trying to have the unique spectral signature which can connect with the remote sensing, with a drought, pest monitoring, climate resilient crop development. Many applications are there. So we envision that sometime back we were writing on notepads. Now we have handheld mobile technology where we are scoring it. And maybe in future, those kind of scoring will be very much automated with AI plus sensors. So coming to the nutrition, I will go quickly because there's like I spend a lot of time on this technology, but this is also important. So one of the thing is like 50 years back, breeding program perspective was different, and now we have difference. So 50 years back, we have a lot of shortage of food. So most of the breeding program only focus on increasing the yield. So yield increment or improvement of crop was the #1 criteria. So they only select for yield. So obvious choice for breeder was to increase the food production, as I mentioned. So what, as a result, today we have 225% increase in the yield compared to 90-60 in wheat. We have 146% increase in rice yield compared to the, let's say, last 50 years. Maize has like 196% increase over the yield. So all the graphs are very, very attractive, very, very motivating. But one question, what happened to nutrition in this last 50 years? I'm sure you and me may not have many example. It's like uh, some piece of information we have on this. But we all know that slowly over time, the nutrition challenge became a big problem. We are seeing nutritional paradox. We are seeing all those malnutrition in children, in women. So this all consequences are happening because over increasing the yield, breeders were having so much pressure to increase the yield. We somehow forget the nutrition. Nutrition was not, when breeder makes selection from F2 to F3 or F3 to F4 or F5, whether they select for nutrition. This was not at all agenda 10 years back or even 5 years back. So this is what, like the some key examples, if you talk about, these are the only examples on your fingertip where some work has been done on nutrition. But these are not part of regular breeding programs. These are the extra work done, and they have never come in a regular practice. They were never adopted by farmers. Those lines which were developed with a high nutrition, maybe their yield is not enough or not good. That adoption at farmer level is not there on this. These are just documented examples. So poor adoption of biofortified varieties and the challenges with breeder, we can understand. The traits are complex. Stability is not there, poor understanding and time consuming process. So if they have to grow 2 seasons per year, they have to wait. If they have 500 lines to do simple protein, which can be done 12 samples per day, they have to wait for 4 or 5 months, whether they can wait this much. So this reason for including not including nutritional trait in their regular selection is obvious. It's very time taking, intensive, and also it's expensive. So it's a time sensitive process and it's also not cost effective. So ICRISAT work on range of the sensors, which is for, so one is called NIRS, near infrared spectroscopy, which is for which work in 400 to 2500 nanometer, and it work for all organic components. When I say organic, it's like protein, starch, the component of nutrition which has carbon, hydrogen, oxygen, and nitrogen in their composition. Then we have an inorganic component. For that, below 400 nanometer spectral information we are utilizing, and that is all X-ray based technology, and it's iron, zinc. All those iron, zinc, and biofortified programs are successful because of this machine. So now ICAR has put a restriction on the release of pearl millet if the 35 ppm of zinc and 42 ppm of iron, if any of your lines is less than that, they will be not eligible for release. So that's why we are deploying this for every season ICRISAT receive 15,000 and more samples across the national partners for screening for iron and zinc. Then we have computed tomography, is a beautiful technology which has a huge potential, and we are using it. So that is another, and also harvest masters. So when breeder harvests their crops, sometimes they are not optimized for moisture. So because of moisture, sometimes wrong assessments are happening where the moisture levels are not equal for all the plots, all the genotypes, and then the assessment yield assessed based on that has a slight variation. So the technology has advancement with that. Like if you go for normal wet lab method, it will take months and months. Then more than a decade, ICRISAT is working on benchtop sensors, which are based on same technology, but they are benchtop. So you have to bring your samples to the lab for this. So that takes few days. But now we are also working on those handheld devices. So this can directly go to field. This can also go to market for quality assessments, or it can it has a potential to trigger your quality-based payment. ENAM, sometime back, government has initiatives on this. So this is very instant in nature. So one of the, if we see the potential, protein 8 samples we can do per shift, per person and per instrument. But NIS can do 250 samples per day in working time. And if you have calibration set for 5 plates, all the 5 plates will be done at the same time. Whether the wet lab, if I have to do fat, then it is only four sample per day I can do. So as I mentioned, this work on spectral technology, which is from 400 to 2500 nanometers. We have XRF, which work for this inorganic, elemental, and compositionals are done with NIRS. One of the example, Girnar 4 and 5, which is high oleic groundnut varieties. It has 80% of oleic acid and less than 20% of non-oleic acid, means linoleic, palmitic, stearic. When I say oleic, it is a good cholesterol, and when I say non-oleic, it's a bad cholesterol. So this Girnar 4 and 5 with the Directorate of Groundnut Research in Junagadh, we work with that, and thousands of samples has been screened. Then this two Girnar 4 and 5 has been made possible to release. If this technology is not there, if those AI-based algorithm calibration models are not there, this might have taken another 10 years to release. So the importance of integrating the technology and at the right time, at the right purpose is very, very important. So this year, half year has only gone, and we have screened 7,000 samples so far for oleic acid. So this kind of technology is giving us advancement, real acceleration in your crop improvement is happening just because of this. Similarly, we are using this NIRS to serve a lot of companies where canola and mustard breeding programs where they want to assess the oil content. If you go outside a Soxhlet method, only 4 samples can be done, and it will take, if you have 500 samples, it will take 70 days with quality control. But if you have NIRS with quality control, like you can do this in 2 days, but then quality check and all those, let's keep another 3 days or 4 days, but still you can deliver in a week time. So this is the advantage we are getting out of this technology and AI. If AI has to be used in right way and right place. This is some of the sorghum germplasm we screened, which are the land races collected before 1981, and exotic collection. We have current breeding lines, and we were able to screen that the new current breeding lines, they are able to increase the protein and fat has been reduced. But all the land races and exotic collections, they were high in fat and low in protein. So we are progressing. We are monitoring through this kind of system where we are seeing that if breeding programs are advancing their protein or nutritional components. We also track different, sorghum has many subspecies. If this high protein lines are coming from any specific geography, because we have germplasm, so there are like some 3,000 lines we have screened. This kind of study is only possible when you have NIRS. Otherwise, it's a huge expenditures. We pay a lot of time in making a good calibration which is accurate enough. We pay special attention to validation, blind validation, normal validation. We do all sort of things so that we know that our models are accurate. If somebody is trusting on this model, this technology, then we have to absolutely assure that your calibration, what has been developed, is very validated. So when I say validation, you build a calibration, you predict the oil content, and you see whenever you use the same sample set on which model is built and you predict, it always have a good R-square correlation. But when you will predict the samples which is not model in model preparation training set is not included, then it will give a check whether it is like it's deviating or how good is the prediction. So we made sure it is above 95% of accuracy. And then we took the independent set, which is like not at all used in that machine. Spectrals are not seen, it's not part of training set, totally blind validation, we say this one, independent set we take, and then you see still has a good accuracy of predictions. So this kind of quality control we are giving to our model so that it predict as accurate as wet lab. Today only we were having a meeting with our companies for which we are giving this kind of predictions, and they said some of our results are better than the local wet lab results. So that also has the chances of machine error, human error, and that gives like more than 10 to 15% of inaccurate results. But then this machine is giving like more than that, like, so they are quite happy with this kind of technology, and they gave a feedback of that. Similarly, for other traits, like it is for protein content. So protein content, whatever model we have, right now it is giving accuracy of more than plus minus 1%. So we are that accurate where we are able to predict the protein content of the samples less than 1% of differences. So with motivation from previous work, we tried building the multiple, always we have to build calibration for each crop, one calibration. So we started building multiple crop, multi-species calibration. So you have one calibration and it can serve many crops. So this kind of models we are developing for our national partners. So if they have machine, many centers have machine, but then they are not using it because calibration is not there, and calibration is the actual skill which is required to serve those machines. So this can be, tech transfer can be done to our national partners. So we are working on this. We also tested in our training set, we did not include wheat. And when we predicted the wheat, like when you include wheat and predict, it's 97%. But when you don't include, then also it is predicting wheat with good accuracy. It's 85%, which is good enough. So this kind of things we are encouraging to work on this kind of, as I mentioned, some of the portable sensors also we are working. They are still advanced, like you can take 300 and more samples with that. This can help the market. India is very decentralized market, and food quality checks are not those up to the mark. So this kind of sensors can help in that. Like some time back, a pilot study under Walmart project we did in Anantapur districts. So to promote our high oleic varieties in Anantapur, we gave the seeds, and we initiated the buyback policy. We gave them seed and asked farmer to grow them and bring it back. And if it is like high oleic, we'll buy on premium price. So first year, all the farmers, they were not totally aware. So they mix with the local variety like K6, and they got it. So whenever this handheld sensors we apply on their sample in different subset, like some 20, 30 subset, and you will see that it will not give 80%. It will not even give 70% of oleic acid. It was giving 40, 50. So you know that it has been mixed with the normal local check. So, so those who grew pure and got the same seeds, they were like paid the premium price. And next year, but farmer got their trust that they know that we can screen it. We know that this is not the original material what we have given them. So next year they all got the pure material. So this is how we can integrate, or we can enhance the nutrition based variety to produce and to feed your value chain so that more nutrition can be served to the end user or the customers. So this was the entry point where we are using. So this is something we also see hope that wet lab, benchtop we saw, handheld we saw. Now maybe at some point of time in standing crop, UVs can also do. Very quickly about CT technology, which is computed tomography technologies. It is again X-ray based. So when X-ray based technology, when you utilize in 3D format, it became a computed tomography. So it is powerful imaging system. As you can see, the groundnut which has been screened, how the filling is there. So pods, we don't shell it. We just use the pods. We scan them, and image-based algorithms are set. Like you see this, maybe two of the pods might be looking same from outside, but the kind of filling they have is very different. So we developed this AI-based algorithms for image analysis. CNN technology has been used, and you do density gradient across that, so you know which is the kernel, which is the pod. And then we developed the algorithm, we predicted it, we validated it, and you see the accuracy is quite good. So with this, now our groundnut breeding program has totally stopped the manual shelling, because for shelling percentage calculation, they have to shell the seeds, and those cannot go to multi-location trials. So they send us the pods, we scan them, we give them four data. One is like shelling percentage, one is kernel weight, one is like the pod weight, and fourth is we are also giving the grade. So grade is very important in groundnut, because bigger the grade, more the commercial value they have. They go for confectionery, and the smaller grades always go for oil extraction. So grade also we are giving, and this is like helping breeding programs a lot. Like, so we don't shell them, we return the pods to them, and the same material go for multi-location testing. So this is how like this. So sensors are always there. Computer tomography is not new, maybe new for agriculture, but for medical industry, it is very common. And how to use the sensors, that AI is helping us. If sensors only capture the images, but AI help us to build a model which can translate these images into data. Similarly, we also work with companies for rice. So some of the traits is very manual. They have to calculate chaffiness, head rice recovery, and it takes a lot of time. So we work with, we have a confidential agreement, so I may not be able to take their names, but we work with them and we develop algorithms. So in one scan, you have all three traits, and this is how it is serving the science. Gene bank, seed viability. Every year they have to spend a lot of time on this assessment of the seed germination test, viability test. So this now we are working. It has been still not optimized, but we started working on viability, whether the embryo viability we can assess. That can save a lot of cost on that. In tur dal recovery ratio we are working, and also plant quarantine unit we are also working on the store grain pest infestations, all those kind of seed health. So final words with this is like technology should intervene the agriculture, my key message. So be open for all the technology AI. Sometimes there is a conservative behaviors on adoption of new technology, but in this era, I think we have to go with AI, with sensors. All the advanced technology are good for agriculture. So we should use it, and to address the climate issue, it is very, very much required. It's a call for the hour. Technology is powerful tool, provided it is hitting the right technique, right research question. So fit for purpose is very important, because you have number of technology. Which one to use, that is important, because always it costs a huge investment. So choose it carefully. It should actually integrate and be a part of regular uses. So those invest on technology, even though if it is small or big, that should serve the like should be regularly used in your breeding program. If it is complex, better to avoid it. So phenotyping platform, what we have developed or chosen it is based on the 15 years of experience of science in enhancing the global capacity for research and breeding of smart crops. So some of the publications are there, and with this, I would like to thank you all. I hope I'm in time. So thank you.
अब इस प्रेजेंटेशन से जो भी पार्टिसिपेंट्स हैं, अगर उनकी कोई क्वेरी है, कोई उनका डाउट है पर्टिकुलर किसी भी एस्पेक्ट पे, आप चैट बॉक्स में टाइप कर सकते हैं। डायरेक्टली आप अपने आप को अनम्यूट करके अपनी डिवाइस को आप पूछ सकते हैं। ठीक है, 10 से 15 मिनट का हम डाउट क्लियरिंग सेशन रखते हैं, इसीलिए कि जो प्रेजेंटेशन रिसोर्स पर्सन ने दिया है, बहुत से डाउट्स होते हैं, काफी क्वेरीज होती हैं, लोग पूछना चाहते हैं, लोग जानना चाहते हैं कि नए नए नवाचारों से हम रूबरू हो, हम समझ सकें उसको। और आप इतने प्रतिष्ठित संस्थान इक्रिसेट में कार्यरत हैं, यदि इक्रिसेट के बारे में कोई जानना चाहे, वहां की गतिविधियों के बारे में जानना चाहे, तो पूछ सकता है।
इंफॉर्मेशन के लिए बता दूं, इक्विसेट हर साल पेड प्रोग्राम कंडक्ट करता है नेशनल पार्टनर्स के लिए, जिसमें ब्रीडिंग के एडवांस टेक्नोलॉजी का ट्रेनिंग और उसके बारे में बताया जाता है। तो अगर कोई इंटरेस्टेड है तो अपना नाम बता सकते हैं।
आपकी तारीफ आने लगी, क्वेश्चंस नहीं है लेकिन तारीफ आ रही। ये बहुत ही इंटरेस्टिंग, बहुत ही ज्ञानवर्धक, बहुत बढ़िया, इस तरीके से जो है आपको कॉम्प्लीमेंट्स मिल रहे हैं।
हेलो मैम।
पार्टिसिपेंट रिक्वेस्ट है कि कृपया आप क्वेश्चंस भी पूछिए प्लीज।
हेलो मैम।
हेलो स्वाति।
हाँ जी मैम। मैम मेरा एक क्वेश्चन है। एक्चुअली बायोफोर्टिफिकेशन इट इज वन ऑफ द टूल to enrich variety or genotype with the nutrients. Okay, but I think so the application of fertilizer and the transformation, both the tools we are using for biofortification. Is there is another tool or technique to study for the biofortification?
So you are using fertilizer, that is the external that is
External application and transformation, both the tools people are using, researchers are using. Any other tool that we can use for the biofortified variety?
See, biofortified, I'm not sure what is your definition, but my definition is like the which has the genetically high increased amount of nutrition. It can be any component, it can be vitamins, it can be protein and all this.
Okay.
जी, नेक्स्ट पार्टिसिपेंट प्लीज।
So until the genotype has the, you can give the standard doses of fertilizer. If you give more than that, also if plant has doesn't have potential to uptake, they will not uptake. So until you improve the genetics, outer management may not help you. So you have to set the high protein line, breed it, and develop the variety.
Okay. Okay, ma'am. Got.
शायद कोई स्टूडेंट इंटर्नशिप के
Thank
बारे
you.
में भी जानकारी कर रहा है।
Thank you, ma'am.
सब हैदराबाद में इंटर्नशिप करना चाहते हैं, कोई कानपुर नहीं आना चाहता, ऐसा क्यों है?
Internship के लिए we are open. We have a LSU system. There is a link also which people can register and express your interest that you want to do internship in this particular area. So the relevant scientist will be contacted once you register, and they can take it in ongoing program if that matching to that interest of the student.
नेक्स्ट प्लीज। हमारे फैकल्टीज और जो साइंटिस्ट हैं, यदि बोलने में कंफर्टेबल नहीं है तो आप लिख सकते हैं, हमारी पर्सनल सेक्रेटरी उसको पढ़ेंगी और वो बात मैडम तक पहुंच जाएगी।
आप हिंदी में भी
एक्चुअली मैम मेरा एक डाउट है कि आफ्टर बीएससी एग्रीकल्चर कोई इंटर्नशिप प्रोग्राम इन आजमगढ़ चालू है क्या?
स्वाति, कैन यू म्यूट?
हाँ, आप प्लीज म्यूट करिए, आपका कोई किड आवाज कर रहा है।
आजमगढ़ में सच तो इक्विसेट का कोई सेंटर नहीं है, और पर एक्सटेंशन प्रोग्राम्स बहुत सारे होते हैं। अभी यूपी गवर्नमेंट के साथ हमारा एग्रीमेंट हुआ है, तो इक्विसेट की कुछ टेक्नोलॉजी यूपी गवर्नमेंट में आएगी, बट दोस आर मोस्टली रिलेटेड टू दिस वाटर हार्वेस्ट एंड लैंडस्केप रीजनरेशन। सो उस एरिया में बहुत ज़्यादा काम होने वाला है अभी यूपी में। वेरी रिसेंटली ये डील हुआ है हमारे डायरेक्टर जनरल और यूपी मिनिस्टर सीएम के साथ। ट्रेनिंग और सपोर्ट प्रोग्राम्स मोस्टली 2 वीक्स 1 वीक का होता है, तो आप इक्विसेट आके अपनी ट्रेनिंग ले सकते हैं और वापस जाके अपने एरिया में इम्प्लीमेंट कर सकते हैं।
एक कोई श्रीमान जी पूछ रहे हैं, उन्होंने चैट बॉक्स में लिखा, नंदिनी इसको पढ़िए प्लीज।
Yes, sir. Very nice presentation. Will there any training program with respect to artificial intelligence in the field of soil science at ICRISET?
Yes, our soil labs are very is accredited internationally, and recently it has started mobile soil lab, which is like a big buses equipped with all those. So they are going each district collecting soil sample, and so there those machines are there which is used for soil, but then they are also trying to use AI for predictions, like if soil, what is the depth, like the soil usually samples collected on the top, but what is the profile, like how much deep it is, what kind of water holding capacity, all this they are using some way for remote sensing, and then prediction of the deeper layer of the soil information. That is one. But second thing also, they are working with this institute which is land use and planning to predict the soil. Indian soils are very variable. If you go to USA and some parts of Africa, that we know that if it is a black soil, the same texture is there for big area like kilometers and kilometers. But India, like every 5 kilometer you have a change in soil. So there they are using AI to predict. So what happen if you have a soil characterized for every 100 kilometer, they're using AI to predict the every 50 kilometer, every 10 kilometers. That some spectral information they capture from remote sensing and some wet lab data, and they are predicting. So those kind of projects are running. Maybe you can be part of that. Like you can write to the relevant scientists. If you go to ICRISAT website, you will find who are the scientists working on soils. You can contact them.
नंदिनी देखिए, लद्दाख से एक डॉक्टर साहब ने क्वेश्चन पूछा है, एक लेडी प्रोफेसर है, उन्होंने एक क्वेश्चन किया है, उसको पढ़िए।
Yes, Professor Jigmit. Any training program on AI in field of soil water conservation, how we can connect?
Yeah, I think we have a whole department of soil conservation, and that was led by Dr. Emil Jad, who is now become a director general of ICAR. So that has a very good program on this. And um, Ladakh, I have visited one. That soil is very different, and you have an environmental lot of constraints there. Um, I, Recently, our team has also visited Jammu and Kashmir. They met CM and governor. I think this is a very good opportunity to work in your area and learn like how on this kind of constraining environment, how this kind of programs can work. Probably if you send me an email, I will connect to the right people.
Thank you, Sunita ma'am. I'm Dr. Jigmit from the question which I have asked regarding this. Now, hello.
Yes, ma'am.
Yes, madam, you are perfectly audible. Please proceed.
तो इस टाइप के ट्रेनिंग लद्दाख के लिए बहुत ये एआई के, अभी तो एआई का जमाना है, तो किस तरह से हम ये अगर इस टाइप के ट्रेनिंग प्रोग्राम अगर लद्दाख जैसे एरिया में आप कर पाए तो इट विल बी हाईली थैंकफुल होगा, क्योंकि बहुत सारे ऐसे अभी स्टूडेंट्स हैं, अभी जितने भी हमारे साइंटिस्ट वगैरह हैं, सब लोग इस चीज को लर्न करने के लिए उत्सुक होंगे। तो अगर इस तरह से कोई ट्रेनिंग हो पाए तो इट विल बी हाईली अप्रिशिएबल। और अदरवाइज हम अगर ट्रेनिंग करना चाहे तो किस तरह से हम एक्लिसेट आ पाएंगे, क्या क्या फॉर्मेलिटीज होंगी, अगर ये चीज आप बता पाएंगे तो हाईली अप्रिशिएट।
हाँ मैम, इक्रिसेट का एक टारगेट ये भी है कि जो एरियाज में ज़्यादा एग्रीकल्चर का डेवलपमेंट नहीं हुआ है, क्योंकि जैसे नॉर्थ ईस्ट स्टेट है, तो इक्रिसेट स्टार्टेड वर्किंग विद असम, मेघालय। मैं खुद भी वहाँ गई थी। तो इक्रिसेट विल बी वेरी हैप्पी टू कम टू योर इन्वायरमेंट, क्योंकि इस एरिया में हमारा ज़्यादा रिसर्च नहीं हुआ है। एक मैं ये कर सकती हूँ कि आपका लिस्ट उस लिस्ट में रख सकती हूँ जो एनसीबीटी ट्रायल्स ट्रेनिंग प्रोग्राम्स होते हैं इक्रिसेट के पेड वन, तो आपको उसमें स्पेंड करने की जरूरत नहीं है। इक्रिसेट विल बॉर्न द कॉस्ट। और दूसरा, हमारे कुछ नोडल ऑफिसर रिसेंटली 1 महीना पहले अलॉट हुए हैं ईच स्टेट के लिए। तो उसमें उन लोगों से आपको कनेक्ट कर सकती हूँ। एक और मुझे मालूम है कि काजरी का कोल्ड डेजर्ट रिसर्च सेंटर कारो में, लद्दाख में कारो कोई जगह है, वहां पास है।
हाँ वो जी 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 मैम
तो हम
जी
काजरी के साथ काफी काम करते हैं, पर शायद कोल्ड डेजर्ट के लिए कुछ भी उतना काम नहीं हुआ है। तो उनके थ्रू भी हम आपको अप्रोच कर सकते हैं और एक प्रोजेक्ट लिख सकते हैं जो इस एरिया के लिए। बहुत सारे डोनर्स आजकल वैसे जगह पे काम करना चाहते हैं जहाँ एक्चुअल में डेवलपमेंट की बहुत ज्यादा जरूरत है। तो ये एक हाईली थर्स्ट एरिया मैं देखती हूँ। मैं आप प्लीज आपका ईमेल अगर अंकुर जी दे दें या मेरा ईमेल आपको दे दें,
मैं दे दूँ। जी
इसको
जी
कोऑर्डिनेट कर सकते हैं इसमें,
जी
इस एरिया में कर सकते हैं।
डॉक्टर अंकुर जी के पास मेरा मेल आईडी सब फोन नंबर वगैरह है मैम।
जी जी
और
जी
मेरा
जी
स्पेशलाइजेशन भी सॉइल वाटर इंजीनियरिंग में है और मैं इस एरिया में काम करना चाहती हूँ। अगर एआई से कुछ इस तरह से होगा तो हम आपके साथ प्रोजेक्ट वगैरह हम कर सकते हैं।
हाँ, तो
डेफिनेटली डॉक्टर।
इसमें मैं आपको कनेक्ट करने वाली हूँ और एक बड़ी टीम है जो काफी काम कर रही है और उनका रिसेंटली कोलेब्रेशन भी बन रहा है। तो आई थिंक हम इसको चैनलाइज कर सकते हैं, ये राइट टाइम है।
जी 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 थैंक यू मैडम, थैंक यू सो मच।
बिल्कुल।
Next question, Dr. Anita. Is there any networking technologies or training program to improve the efficiency, safety, and traceability of GM food processing, genetically modified foods, or their processing?
नहीं, एक्चुअली मेरा एक्सपर्टीज इसमें नहीं है। I'm not sure on this because in this area I have not worked much. There is a editing team here, but I'm not fully aware how much they are on this area, traceability.
Okay. Next, please.
From Kaushik Singha, if any agricultural startup farmer community intend to contact for tech-related guidance or support from your organization, then how to take opportunity from your organization?
So we have a Agri Incubation Center here, AIB. They incubate startups, and they also support and they connect with ongoing research. So that and we also do tech transfer. So ICRISAT is a non-profitable organization. We develop technology, but we don't look for profit. So we do a tech transfer and initial handholding for the startups to extend those technology and imply and develop a business case. So they can contact AIP or incubation centers if they like to be incubating in that ICRISAT. They can choose any of this mode to contact to associate with ICRISAT.
Okay, I want to learn AI and image processing for plant disease detection. How can I get connected and learn on it? Dr. Ritesh.
Yeah, so we are not IT institutes, but now a lot of training programs, external training programs are happening where they're training for disease detection. One of the Plantix app, which has been developed with ICRISAT collaboration with one of the European company, that has worked very well, and it is AI based, and also it has strengthened because of crowdsourcing. Means people were sending the pictures and those were still used in training. So crowdsourcing help you to strengthen your model. So we have a digital technology department. There they can contact and be part of that disease detection. But as such, we are biologists. We don't give training for AI. AI training, you have to like basic running the models, writing the codes, application of those automosaic, all those kind of things. You have to learn from any IT engineering courses, professors or private training organization. And then on application part, you can work with ICRISAT or any organization whom you trust, like, or you would like to work, interested to work with them.
Madam, good evening.
Yes, sir.
I'm Dr. Ritesh from Centurion University, Odisha.
Yes,
So
a lot
I
of
want-
students are coming from Centurion University for internship at ICRISAT.
Yes, I know. These all are our BSc AG students. This year also, I think 4 of them have went there.
Yes.
So I just want to know from you, how reliable is this plant disease detection with the help of image processing? Whatever I got to know that there are so many complexities. We cannot rely on the result which is generated by image processing because of the symptoms which are very confusing to each other. So image processing, just I want to know how much reliable it is.
It depends your training set on which the model is built. If model is built with over 30 training set data, it will be not at all reliable. But if your model is built over 30 lakhs training images, then you can rely on it. So how robust is the model? That is one thing we have to ensure. That's what I was emphasizing. AI is very dynamic. It can be worse than your normal statistical model, and it can be at the best or the magical of those so far
Yes.
what technology advancement we have seen. So always go for the applications which is built over large set of data set. With the small set of data set, you will always have a risk. And I will tell you that a lot of startups they build over a small set of data set, and then this doesn't work.
Yeah, that's why they are not reliable. Yeah. So data set should be strong.
Yeah, very strong. Like it has to be very. So those Plantix, because crowdsourcing has been used in that, so it is built over like I don't know, more than 10 lakhs training data sets. So that the predictions are approximately accurate, but still I will say there is also chance of error in that.
Yes, it is. It is there in Plantix. Errors are there. Okay.
Yes, error is there.
Thank you, madam.
So last question, is there any training program for agricultural economics?
We have a department. I'm not sure if those training are regular. For
So
breeding,
that's
most of our technology or training is around those breeding. So national partner breeders are more than welcome for those all trainings.
so Ankur sir, that was all the questions. If still anyone left, you can still ask. Otherwise, we are winding up the session.
बस ठीक है, अब धन्यवाद ज्ञापन करते हैं.
So, on the behalf of entire AgriMeet Foundation Bharat and all the participants, I sincerely thank you, ma'am, for joining with us today and delivering such an inspirational session on AI technologies for accelerated crop improvement and climate resilience. Your clarity, passion, and depth of knowledge made the topic not only easy to understand, but also very motivating for all of us. The way you connected innovation with agriculture truly encouraged us to think differently and reminded us that learning has no age. Ma'am, your presence added immense value to this faculty development program, and we are truly grateful for the time and wisdom you shared with us. Thank you once again for being with us today.
Thank you to you and also the audience. Thank you. Bye-bye.
Everyone can leave. The session is over.